Hello, my name is KayLee Ridge, and I am a junior biology major at the University of Evansville in Indiana. And this summer here at DISL, I have been characterizing the necrobiome of bottlenose dolphins in using 3 variables: individual variation, age class, and condition code, as well as examining the efficiency of collecting the necrobiome by examining skin tissue and skin swabs. So to better understand the necrobiome, we need to understand the microbiome. So the microbiome is found inside and outside of all living organisms and can provide important information about the health of the organism as well as potential threats to their population. They can also act as a protective barrier on the skin to shield any organisms from pathogen overgrowth or just any pathogens trying to get in. At death, the external microbiome is then called a necrobiome, which is believed to contribute significantly to decomposition of these organisms. So our past REU student collected our data for us where we performed the bioinformatics because she performed the pilot study that leads up to my study. But since my sequencing and bioinformatics is not back yet, I did her bioinformatics. So she collected samples from 10 deceased bottlenose dolphins from 2020 to 2021. These are the sample demographics. Here you can see the age class, and then how many of each age class we have, and then our conditions codes, and then how many vary between all of our samples. So from these dolphins, we collected 1 dorsal skin tissue, and then 1 dorsal skin swab from each dolphin. And then we performed a DNA extraction and sequenced them. So our results that we found, the 6 most abundant phyla found on our dolphins are seen here. Here we have the protobacteria, which is the orange, and then the firmicutes, which is that pinkish color. Those are the 2 most prominent phyla found, and those are often very common found on bottlenose dolphins, which leads us to believe that these are possibly dolphin-specific microbiomes or phyla. So down here we have our alpha diversity. So here's our sample ID and then the richness number of ASVs, and ASVs are just proxy to bacterial species. And in the pink we have the swabs, and then in the blue we have the skin. And here just shows us that the skin swabs yielded a greater richness than the skin tissue samples. And then down here we have our beta diversity. So dolphin ID, code, age class, and age class affected the microbiome. And here we can see this with our dissimilarity plots. So the further away the plot or the dots are, the more dissimilar they are, and the closer they are, the more similar they are. So here we can see that our code 2s, 3s, and then the early code 3s, they're all kind of clustering with their own codes. So this can show us that there possibly is a correlation between the necrobiome and the codes, as well as age classes, where here we can see that the age classes are also clustering, showing us that there may be a correlation. And then with our individual dolphins, we can see that they are all kind of far away from each other, or at least distanced from each other, showing us that each individual dolphin has their own necrobiome, like an underlying necrobiome system. So overall, we found that bacterial richness showed a difference between skin tissue and swab samples. This finding could be due to 2 different reasons. One reason being that the skin samples had more dolphin DNA, making the bacteria harder to extract, or that during collection the swabs covered more surface area, resulting in a greater yield. And as a result, we recommend using swabs over skin samples. As well, we found that dolphin ID, decomposition code, and age class had the most significant effects on the bacterial community composition. So the necrobiome differed with decomposition code, which may be promising for determining the time of death with additional study of all the other factors. So overall, the results suggest that every organism has an individual community, making the microbial community differ largely between individuals as well. That's it.